Hello, listeners. Here we are on episode 3 of the Immunology for Everyone podcast by SKA. Your host and subject matter expert, Professor Dr. Sunil K Arora, is putting together his knowledge and vast experience in the field of immunology for all of us who are keen on learning. His subject matter proficiency comes seasoned with clinical collaborations and research that has been dedicated to improving diagnosis and discovering better therapeutic targets, taking us step by step into understanding immunology from the very basics to its applications. He is here today to tell us about our immune system in a way it has not been told before. So get your earpieces on and gather every bit he has to share. Queries are very welcome in the comments and will be responded to by the expert himself. Keep listening, liking, and sharing these podcasts. Here is SK for you today.
Thank you very much. Once again, hello everyone. Hope the last podcast where we discussed the becoming of immunology must have opened some gates of interest in your minds. And maybe now we can formally begin to recognize the individual components of immunity that actually play the real game against all the pathogens and foreign agents that invade our body. We now very well understand that in order to address the threat of pathogenic microorganisms, we have evolved an immune system comprising of network of specialized cells, tissues, and molecules. A part of this system is innate to our body, as we are all born with this. The mammals have inherited this system from invertebrates during evolution. So while the other part of the immune system is called the adaptive immune system, as it evolved through vertebrates to generate an antigen-specific response. So this means that the innate immune system, which is more ancient, is comprised of mechanisms that do not involve antigen-specific responses and so do not generate any memory, which is the hallmark of adaptive immune response. It is also clearly understood now that these responses are not dependent on prior exposure of specific pathogens for their amplification, as they are controlled by the products of germline genes that are similarly expressed by all the living objects, from most primitive single-celled organisms to higher vertebrates in the evolutionary tree. So the immune system, which is innate immune system, it involves both constitutive and inducible components, and it uses a wide variety of recognition and effector mechanisms. The research have now made it very clear that the innate immune responses have a profound influence on the generation and outcome of the adaptive immune response. So it makes it very important to understand the concepts of innate immune response before we understand the generation of pathogen-specific immune responses. So today, I plan to talk about the main components of this intricate system that provides the first line of defense. So in order to provide protection against wide array of pathogens, the innate immune system has evolved to include two-pronged defense components. Some of these are external and the others are internal, which complement each other whenever they are functioning. So the external components, they include the anatomical or physical barriers, some effector cells, some antimicrobial peptides and soluble factors, and also the receptors which are present on these cells. So let us go one by one. The most prominent among barriers are the skin and mucosa. These provide an effective anatomical barrier between the internal and the external environment of our body, as they block the entry of pathogens into the organs. So the skin, which covers our body all around, acts not only as a physical barrier, but also as a chemical shield. It is made up of three layers: the epidermis, dermis, and hypodermis. And all these three vary significantly in their anatomy and function. So the structure of skin is made up of an intricate network which serves as the body's initial barrier against pathogens. It also protects us from UV light exposure, some chemicals, and the mechanical injury. So the most external layer of skin is epidermis, which is mainly consists of some cells which are called keratinocytes. These are very tightly linked by desmosomes and are embedded in a layer of extracellular matrix proteins. So these keratinocytes, they also express certain receptors on their surface, which are called the pattern recognition receptors, or the PRRs, because these can recognize and bind to some molecular patterns which are commonly expressed by groups of pathogens. And these molecular patterns are also called pathogen-associated molecular patterns, or the PAMPs. We'll be talking about PRRs and PAMPs quite frequently in the next few podcasts when we are talking about the activation and generation of both innate as well as adaptive immune responses. So on this interaction, the keratinocytes produce many cytokines, and these cytokines are normally the soluble mediators which are released by one cell, and they go and bind to another cell where there is a receptor for this cytokine, and they carry a message from one cell to another. So these cytokines, basically, when they are released, they induce some kind of reactions or activation cascade, which also include the inflammatory cascade. So the keratinocytes also produce many antimicrobial peptides besides these cytokines, and these antimicrobial peptides, they cause the destruction of microbes. So in addition to these keratinocytes and the secretions, we do have certain glands which are called the sebaceous glands. These glands are associated with the hair follicles in the skin, and these sebaceous glands, they produce many fatty acids, and they create a acidic environment on the skin, which is basically lethal to the bacteria which settle on the skin. So you can imagine that any malfunction in the epidermis can lead to inadequate host response to a pathogen or a persistent inflammatory state. So here I give you an example of a very common inflammatory skin disorder, which is kind of dry and itchy skin, also called the eczema or atopic dermatitis in medical terms. This condition can occur because of a combination of things. It may happen in few individuals because their skin is unable to retain much moisture. This dryness may make the skin more likely to react to certain triggers, like some irritants, which can include the soaps, shampoos, or environmental allergens like dust mites, pollens, etc., and some fabrics also, like wool or some synthetic clothes like nylon or something. So although there are this kind of condition is more common in children who have a family history, but it can be observed in any age. So the main cause of this condition is the abnormalities in the skin barrier structure, and which cannot trigger the clearance of pathogens, and which means that there is a defect in the innate immunity and an altered microflora of the skin. So this is a irritating disease, but it is non-contagious because it is not infectious in nature. So many other abnormalities may occur as a consequence of epidermal dysfunction, through pattern recognition receptors. So then I talked about another anatomical barrier, which is also very important, which is called the mucous membrane or the mucosa. This mucosa lines the insides of organs and cavities throughout our body, and that are exposed to foreign particles from various from the outside. And all these openings of mouth, anus, ears and eyes and all, nose. So all these openings, and the tracts are actually covered with the mucous membrane. So this creates a very sticky material which is called mucus. So the mucus secretions, they lubricate and protects these organs and cavities from abrasive particles and invasive pathogens. The epithelial cells in the gastrointestinal and respiratory tract, the mucosa have certain kinds of hair-like structure which are called cilia. And once the function of these cilia, they keep the moving and they clear the mucosa of mucus dust and also possible invading microorganism which get stuck to it because of the sticky nature of the mucosal membrane. So besides these skin and mucosa, we have a very important, anatomical barrier, which is known as the microbiota or the normal microflora of our body. It is amazing to know that more than 1,000 species of microorganisms, which may be comprising about 10 to 100 trillion symbiotic microbial cells, which are harbored by our body. And they are mainly in the skin, in the gut, but also on the skin and mucous membranes. These bacteria collectively are known as microbiota, and they start colonizing our system mainly in the gut, also in the skin and mucosa, immediately after the birth. Because the child is taking breast milk, a lot of bacteria, they go from there and colonize in the gut. The food we eat, the home where we live and the environment, the contact with many people who visit us, the pets we have. So everything affects the diversity of this microflora. So most of the scientific data, it reveals that it is healthy to have more diverse kind of microbiota, as it can influence not only the outcome of our immune system to any infection or cancer, but also in general, it can influence the mental health and other general health of our body. So these bacteria provide a strong anatomical barrier because they compete for food and space with the pathogenic organisms, and they prevent their growth, and also they help in the normal functioning of our system. It is important to know that any disturbance in the population of these bacteria may be due to, for example, if you're taking some broad spectrum antibiotics or for a prolonged period, then you might have observed that some people, they get diarrhea. And diarrhea is because of the disturbance in the microbiota of the gut. And then if you eat a lot of curd or yogurt, or maybe take probiotics to replenish them, and that actually restores the normal functioning also. So it is very important to know that this forms a very, very important component of our body system, which influences the immune response also. So besides the anatomical barrier role of skin and mucosa, there are many secretions, like we have sweat, we have saliva, tears, nasal secretions, gastric secretions. They all have an important role to prevent the entry of pathogens into our circulation. These secretions, beside playing a flushing action, like you have, if something goes into the eye, there are tears, they flush it out, and similarly nasal secretions. So flushing action is one functional component, it also contains several chemicals and some enzymes which have the antimicrobial action. So if we look at what are these components, there are certain enzymes like lysozyme, which is present in the saliva, tears, and nasal secretions. Then you have hydrochloric acid and digestive proteins in the gastric secretions. The hydrochloric acid is highly acidic, and the acidic atmosphere is not good for pathogens and they die. And then digestive proteins like pancreatin and peptidase, they are also in the gastric secretions in the GI tract. And then you have the fatty acids and the bile acids, you must have heard. These all, components, they basically are secreted in the gut, and they protect us from the pathogenic organisms because of their microbicidal activity. So there are many mechanisms that initially try to prevent the entry of pathogens by blocking and then killing action even before the pathogen could gain the entry into our circulation. These all form the first line defense of innate immune system. So we can envisage that any breach in the barriers or the secretory system may allow the entry of some pathogens, which may subsequently cause the disease. And as I previously mentioned, that many of these cells of the skin and mucosa, they have certain pattern recognition receptors, which are expressed on the surface of these cells, which can bind to the pathogens and molecular patterns. There are certain, soluble PRRs also. One of these is the mannose binding lectin, and also the C-reactive protein. The C-reactive protein is an acute phase protein produced by liver and secreted into the blood. It is often the first evidence of inflammation or an infection in the body. Whereas the MBL, which is the mannose binding lectin, it can recognize the carbohydrates on the microbial cell walls and acts as a opsonin. Opsonins are kind of those molecules which can coat the surface of the bacteria, and when they coat, they are basically phagocytosed by the cells of the system, phagocytic cells. So it is a kind of facilitated phagocytosis and the complement activation for quick removal of these pathogens. So when I mention the complement activation here, so I may like to tell you that there are several soluble protein factors of our system that are called the complement system, which are circulating in an inactive form in the plasma of all the healthy individuals. Once a pathogen enters the body, it triggers an activation cascade leading to the activation of the complement, which is kind of chain reaction, with one activated component leading to the generation of another activated component, and so on till the formation of an membrane attack complex. This membrane attack complex, also called the MAC, it basically attaches to the surface of the pathogen on the cell wall or cell membrane, and literally it can create a hole, many holes on the walls, which leads to the release of intracellular components, as well as bursting of the pathogen or that cell, which kills the pathogen. So complement activation pathway is a very important component of our innate immune response also. There are three mainly pathways of activation of component. There is a classical pathway which is mainly triggered due to the antigen-antibody complex. Since we have not talked about antibodies yet, so I will take up in some other, subsequent podcasts when we are already done with the antibody formation and complex formation. But there is another pathway which is called the alternative pathway of complement system, which is an innate component of the immune system's natural defense against infection. So this pathway is activated by the viruses, fungi, bacteria, parasites, cobra venom, immunoglobulin A, and polysaccharides, and form an important part of the defense mechanism, which is independent of the immune system response, and it can provide the activation of complement and the protection against these pathogens. And there is a third pathway, which I already mentioned, the mannose binding lectin pathway, which is basically, I told you, it can go and bind and trigger the activation of complement. So the complement system is also a major component of our innate immune system and gives a initial protection from the infecting agents. So besides this complement system, there is another very important system that is involved in the innate, protective mechanisms, and that is the coagulation system. And you must be knowing that the normal function of coagulation system is to control bleeding and clot formation during any tissue injury. But this also provides the microbial dissemination, prevent the microbial dissemination, because a component of this system, which is called fibrinogen, it can go and bind to the pathogens. And once it does that, because it senses the microorganisms, and it acts as a opsonin, which I told you that the coating, and then facilitated phagocytosis for clearing these parasites from this surface. So that also provides a very important, protective mechanism of the innate immune system. So apart from all these anatomical barriers, the innate immune system has a battery of effector cells. These cells, so once there is a breach, there is a pathogen entry, then these bacterial cells, which are present in the mucosa and the skin and all the other organs, which are peripheral organs, where the exposure is more common, and these form the frontal force for elimination of invaded pathogen. So these cells are the phagocytic cells, which consist of the granulocytes like neutrophils, eosinophils, basophils, and mast cells. They all have their own action. Some of them, they act as phagocytic cells. The other cells, they act as in the allergy and all. So we will discuss their roles subsequently in next podcast. You have monocyte, macrophages, and dendritic cells. They all have phagocytic activity, and once the pathogen is phagocytosed, then a number of effector mechanisms they are basically triggered, which can kill the intracellular pathogens. So the number of these cells you might have seen in your blood reports, and you can imagine any decrease or increase in these numbers, it signifies the immune activity against pathogens, and the doctors can correlate with the type of infection. Sometimes some of the components, they are increased in the bacterial infections, while others are disturbed because of the viral infection. So it's basically giving them the suspicion so that they can go and become more specific to start the treatment. Then besides these phagocytic cells, there are other cells. You have the epithelial and endothelial cells. There are natural killer cells. We have the plasmacytoid dendritic cells and innate lymphoid cells, as well as the platelets. These all cells, they also form the cellular components of innate immunity. And I plan to take up their specific functions of some of these cells which, in the next podcast, in the coming up next. So majority of these cells, they also express the pattern recognition receptors, some of which are extracellular, DPRRs, and some are in the cytosolic as well as in the endosome also. So which include the toll-like receptors, the nod-like receptors, the rig-like receptors, the cytosolic DNA sensors. So all these receptors, they are called the pattern recognition receptors, and they recognize various components of different pathogens, and which we collectively call the pattern recognition receptors. So on recognition of the pathogen or the molecular patterns, these cells, they secrete many kinds of soluble mediators, which we call these cytokines, to initiate a cascade or a phenomena which is called the inflammation, and also triggering the antimicrobial effector mechanisms. So I'll be brief with the antimicrobial mechanisms today, because we are going to take up them in the details in the next one. But for your knowledge, there are production of reactive oxygen species, reactive intermediate, oxygen intermediates, because of certain enzymatic complexes, and then oxygen independent mechanisms, because of certain enzymes like proteases, cationic proteins, lysozymes, elastases, cathepsin G, and defensins, etc. Everything has all of these have the microbicidal functions. And then in addition to all these, there are certain antimicrobial peptides, as well as reactive nitrogen intermediates, which are produced by the neutrophils and other cells, and also the DNA extracellular traps. So we will discuss about all in a little bit more details to understand their function in the next one. So we see that there are important mechanisms that play a role to prevent the entry of pathogens into our body through physical barriers, as well as the antimicrobial action of factors which are released by cells which are associated with these tissues. So all these mechanisms, they comprise the innate system of our body. The cells of innate system also play a very important role in providing protection against the onslaught of any infectious agent or even the cancer cells. And so I briefly talk about those specific functions. And before I close, I would like to have you enlist some of the disorders which you might have heard or know of, which may relate to the, in particular to the defects in the innate system. So you may enter your responses in the comment box after this podcast, and we will see if they are included in the next one also. So looking forward to meeting you soon. Till then, thanks and goodbye.
Thank you, Professor, for this introduction to the body's first line of defense. I have very limited time for just one question that I have already received, if you can take it, and the others will be taken up in the comments as they come in. SKA, the question is, why do different people respond differently to certain pathogens? Like COVID-19 was a new virus, and still there is difference in symptom severity or even the symptoms among people. Why is that?
Well, it's a very, very interesting question and quite relevant here. You see, I'll take the example of this COVID-19 pandemic only, which is caused, the COVID-19 disease is caused by the infection of a virus, which is called SARS-CoV-2. So, and you have seen recently there is a, spectrum of response by the general public. So we saw that during the peak COVID-19 season, when everybody was kind of getting infected, still we found that more than 80-85% of people would not even have any symptoms or will have very mild symptoms, but they recover very fast. And they recover within a few days, like even before the actual adaptive antigen-specific or virus-specific immune responses could have generated. Because normally the antigen-specific immune responses will take about 5 to 7 days to set in and generate the specific antibodies which will go and bind. So you have heard about the neutralizing antibodies and T-cell activation and all that. That happens a bit later. So you can imagine how majority of these They get away with very mild symptoms or maybe very short-lived disease. That all is because of the innate immune responses, which we discussed today. So it happens with many infections, almost all, because every infection, many people have, the robust innate immune system, which can take care of the initial part of it. And many of the infections are cleared even before the triggering, even before the generation of specific. Although the innate immune response goes on to activate the adaptive immune response, which is more specific, very specific against the same pathogen, and that we will come to know in the subsequent episodes when we discuss about the further activation of the immune.
All right. I think quickly we can take another one in a minute or two, SK, if you have. Uh, are there any disorders or diseases that happen due to any defect in the innate immune system, that is from birth or genetic defects?
Very nice question again, and I have already asked this response from the listeners. But to mention here a couple of things, I talked about uh, MBL, the mannose binding lectin, which can trigger the complement activation also. So there is a very important disorder which basically occurs because of the inborn error of immunity, because of the deficiency of this MBL, which is because a defect in the MBL2 gene which expresses the protein. So this particular deficiency can be found in about 5 to 10% of the population world over. So this is basically because of the defect of a gene, which basically makes the component of an innate immune response. That is one example. And there is another example of this, which is basically I talked about, generation of, although it is premature, in the next one I will take up, but the production of reactive nitrogen species, the reactive oxygen species, because of the activation of certain enzyme complex. One of the enzyme complexes NADPH oxidase, which is also called NOX2, and a deficiency of NOX2, which is kind of again inherited deficiency, can lead to a very this kind of disease, which is called the chronic granulomatous disease, or CGD. Although it is a very rare inherited immune deficiency, one in say 200,000 or one in 250,000, you find it, and it results in the inability of the phagocytes to generate enough of superoxide ions, which are the oxygen intermediates. They are lethal. So if that is not properly produced, they also cause this kind of recurrent, life-threatening bacterial infections or fungal infections and chronic inflammatory disease. So these are two very important examples of inherited disorders, and we categorize them into, inborn errors of immunity or the primary immune deficiency diseases.
Thank you. That was perfect. And until we meet next time to hear you further on the cellular components, thank you, everyone.